Okay, welcome everyone to the shortly before hibernating winter session. Today our speaker is Priya Khanna. Priya is one of our own. She got her PhD here a little while ago, and she then went to do a postdoc at UCSF, and then came to us to be a professor here. And Priya is an expert on the interaction of motion and sensing, if we sort of very broadly describe it, in the brain, working on neuroscience, network science, and neurotechnology. She has several awards. She has a Sloan Fellow, she has an NIH, New Innovation Award for work she's going to talk about here. She has a Google Faculty Research Award. She was a young investigator for the Simian Collective, just to name a few. I don't want to go back to her PhD times. And she is going to talk today how we can map and mend dexterous movement control with neurotechnology. Welcome, Priya.
Thanks. All right. Thank you for the kind introduction. So we have a pretty small group here today, so feel free to move forward. Feel free to interrupt me. Happy to make this as casual as possible, and feel free to ask. I can go into more depth for a lot of this. This is going to be a higher level overview. So I'm really excited to be here today. My group is a new lab at the intersection of the new neuroscience department at Berkeley, so you can see their logo there on the right, or left, your left, and EECS. And we just launched in summer of 2023. And so today I'll be talking at a very high level about some of the problems we're going after and some of my previous work in tackling these problems and new directions that we're trying to take in the lab. So my big picture interest is in developing models of the very commonplace yet remarkably fast and flexible dexterous movements that you and I do every day. And then in trying to leverage these models to engineer neural interface therapies to restore upper limb function in disabled patients. So just to orient us, the patient population that I'm going to be focusing on the most today are stroke patients, since it's the leading cause of long-term disability. There are about 800,000 strokes per year in the US, and about 50% of stroke survivors exhibit disability even after physical therapy. So to give you a sense of what I'm talking about when I talk about upper limb disability, I'm just going to show you a couple of videos here of data that I collected in my postdoc at UCSF. So this is a patient with a unilateral lesion on their left hemisphere that ends up influencing the movement of their right arm. So first, I'm going to show you them doing a movement with their arm, with their unaffected left limb. So what you're going to see is the person reaching out, grabbing an object, releasing it, and then repeating that 10 times in a row. So you can see that this movement is really fast, it's fluid, it's smooth, it's something like what you or I would do. And it's really repeatable from trial to trial. And so then if we look at the other limb, the impaired limb on the right side, you're going to see a really different picture. So this movement is now a lot slower. There's tremor, there's instability at different points in the task. There's quite a bit of difficulty releasing the fingers and releasing the object. And you can see that there's a lot of inconsistency from trial to trial as well. And so if we take a coronal slice of the brain and look at the pathways that all affect movement, we can start to understand why there is such multimodality in symptoms, as well as why this upper limb disability has been so hard to treat. So what I've highlighted here are just a few of the pathways that we know are really critical for movement control. So the purple pathway is a canonical cortical spinal pathway. So this takes information from the cortex or the brain and travels down to the spinal cord to animate the muscles. But in addition to this pathway, we know there's also information that's being passed from subcortical nuclei, like the basal ganglia, thalamus to cortex. These are shown here in blue and in orange, cortico-cortico pathways, and then colossal fibers that help hemispheres coordinate the different limbs. And so the reason I'm showing you all this is not to throw a bunch of jargon out about systems neuroscience, but really to highlight that strokes tend to occur at the intersection of all of these different pathways, which makes it really challenging to try and disentangle what movement deficits are in individuals. And so what's shown here is where the most strokes tend to occur, which is at this intermediate phase between the subcortical and the cortical nuclei, and really at the intersection of all of these different pathways here. So hopefully this gives you an appreciation for the complexity of the problem that we're trying to go after. So,
Talk about bleeding strokes or about blocking strokes?
either, yeah, wherever there's kind of localized damage. Yeah. And so one type of device that I'm really optimistic about are what I call neural interface devices. So within this class, there are neuromodulation devices. That's what I'm picturing here on the left. So these are devices that are either implanted or on the surface of the body and inject small amounts of electricity to bias the firing of networks of neurons. So a classic example is the deep brain stimulator that was developed for Parkinson's disease, like you see here. But since the 1970s, when this is developed, there's been a huge technology explosion in this area. So there are now devices that have multiple different form factors and implant locations and treat kind of a diversity of different conditions. So this is a vagus nerve stimulator that treats epilepsy. There are TMS coils that treat depression. There are wearable devices on the wrist that manage essential tremor, and the list goes on and on. In addition, one of the reasons I'm so optimistic about these devices are that they have historically been used to treat movement disorders, so things like Parkinson's disease, essential tremor, dystonias. These are all managed well with neuromodulation devices. Second, neuromodulation can operate at the same timescale as movement. So unlike a drug that might take minutes or hours to wash into the system and wash out of the system, these devices can be programmed to give very fast pulses on the order of tens to hundreds of milliseconds, which is the timescale at which we move. In addition, they can be really precisely targeted. So in the last slide, I showed you that people can have really different patterns of damage. And so these devices can be localized to particular pathways that might need to be strengthened. And they can be personalized. So these devices can be responsive to different sensors that we can place on the body or on or inside the body. And so speaking of sensing, there's also been huge developments in sensing in the last decades. So this was one of the first intracortical electrode recording arrays that was super bulky and stiff, and now that's been really ousted by some of these more sleek technologies that have flexible electrodes that can go inside the brain and record and wirelessly transmit that outside the brain. There have also been innovations in monitoring muscle activity and behavioral monitoring as well, from things like the Apple Watch or from camera signals. And so all that I'm trying to convey in this area is that we have a lot of technology available to us as clinical neuroscientists or systems neuroscientists or neuroengineers. And what's really astonishing to me, at least, is that currently there's no device that's part of standard clinical care for upper limb disability. And so where is the problem? Why are we having difficulty in applying these devices? So this is where we are right now. Now what I want to talk about is where I think we can be in a couple of decades. So what I imagine is that someone would actually be able to come into a clinic, they might have a brain injury, and we're able to do a battery of tests. We're able to look at their neural signals, their muscle signals, their behavioral signals, and we're able to have such a good understanding of the physiology that we can actually identify what types of neural signatures are missing or what types of neural signatures are exacerbated. And we can combine that with neuroimaging data or patient history data and actually make a physiology-informed prescription about what types of devices might be really appropriate for this particular person. And so instead of having one device, we can have a whole suite or buffet of devices that we might be able to choose from depending on what deficits a person has. So maybe someone is still really excellent at motor learning and motor control, but they just have weakness in a subset of muscles. Maybe they would benefit from a wearable robotic system that provides support to those muscles. Maybe they have a tremor that's really difficult to manage. Then we can prescribe them with something like a peripheral nerve stimulator. Or maybe they have certain types of neural activity that need boosting or suppressing. Then we can try and prescribe something more at the central nervous system level. And so we're very far away from this, but what I hope we can do is actually be able to get towards this as we perform research in two main areas. So this vision has really shaped what my lab is trying to go after, which is two main goals. So the first is advancing our systems neuroscience models of fast, flexible, and successful movement, both in intact networks and then seeing how these models break down in impaired networks and movement disorders. And then second, in engineering physiology-grounded therapies, so actually understanding how these interventions into the nervous system are shaping and modifying the physiology of the system to be able to give back some of the neural signals that might be missing or suppress some of the exacerbated signals that might be too much there in an impaired nervous system. So these are the two categories of research directions that I'm going to be talking about in my talk going forward. So if we start with advancing models, before we jump into thinking about how the brain actually controls movement, a really important factor to consider is what is actually good movement and what is impaired movement. So if we look at this video again, this was the right arm from that first slide that I showed, you and I can look at it and we can say, yeah, that does not look right. Something is not quite right there. The movement is slow, it's tremorous, but what actually is the language that people use to describe this type of movement? So if we ask the clinicians, or researcher clinicians, they'll tell you that there are a battery of clinical scores that can be used to describe this movement. And so these scores are really useful. They're a very low dimensional representation of the movement impairment. So this is a very common one used in stroke and upper limb disability is the Action Research Arm Test. So that's a test which tests gripping, grasping, and timed functional behaviors. And it spits out a score from 0 to 57. And so this participant here is a 34. So we know that they have some impairment. They're not a 57, which is what you or I would be. And they're not totally paralyzed, which is what a 0 would be. But it doesn't tell us very much about the qualitative aspects that you or I could identify. So it doesn't tell us that the movement is slow or tremorous or there's a lack of coordination.
Can you tell the difference between a stroke and an asthmatic hand, not asthmatic, arthritic hand?
Arthritic hand.
Yes.
I mean, the ARAT cannot. So this is a very functional outcome. So it tells you, can you do certain types of movements and how many of those movements can you actually accomplish?
So if somebody is in their 90s and has an arthritic hand, they will not have a 57?
Probably not. It would probably take them longer to do certain functions, or they might not be able to do certain functions. So yeah, it's not specific to stroke. Yeah. And specifically, it's also not specific to what joints are impaired in stroke. So it's possible, as I mentioned before, that upper limb function or kind of the arm function is really successful, but it's hand function that's impaired or vice versa. And so it doesn't tell us the difference between these. And that makes sense. It's clinicians need kind of a fast understanding of the deficits of patients. But the problem comes when we're trying to yeah, go ahead.
How do you give or take away points in this score? Like, how do you arrive at that 34 exactly in this?
Yeah. So there are a few different categories, each type, and within each category of movement, so categories are gripping, grasping, and within each category there are a few different types of movements that you do. So for example, for the grasping, you can reach out and pick up a cricket ball or a tennis ball. If you do that, and you do that successfully within a certain time limit, you get, I believe it's a 3. If you do it and it takes you much longer than that, you get a 2. If you can't do it, but you can try, you get a 1. If you can't do it at all, it's a 0. So it's very qualitative, kind of based on timing and success of the movement.
Is it a computer that's giving these scores, or is it by just a human watching the trial?
Yeah, that's a great point. It's a human watching the trial. So this one actually has quite high inter-rater reliability, but some of them don't. Yeah. So yeah, you bring up a lot of great points, which is that this is clearly a representation of the person's movement that can be useful, but it's missing a lot, especially if we're trying to think about what types of movements are impaired and how might therapeutics interface with physiology.
But you don't get just the score, you get the scores on each subtask, right? Which will tell you a little more.
Yeah, if you looked at that, yeah. So one of the things that you can think about doing is monitoring kinematics and actually monitoring these joint angles in more detail. So myself and a colleague developed a system to be able to do that and developed a standardized task which allows people to reach and grab objects in a way that it allows us to monitor very easily. And so one thing we did then is we recruited participants that actually all had very similar ARAT scores. So I know this is a little small, but this is a person with an ARAT of 38, 34, and 36. And so they're all within a couple of points of each other. And then we zoomed in and we looked at some of their movements. So this is the movement of the thumb IP. So this is your thumb joint here. And what I'm plotting here are 10 trials of movement for both the stroke-impaired participants as well as the neurologically intact participants in black. And so what you can appreciate is that there are really different patterns of dysfunction for these different participants. So in black, you're seeing the neurologically intact participant has about a flexion of about 20 degrees. this participant here on some trials has no thumb flexion, on other trials has thumb flexion that is about 20 degrees, but there's a lot of variability from trial to trial. This participant has, it seems like an uncontrolled tremor, and so there's very little flexion above that baseline tremor. And this participant has flexion that's fairly consistent from trial to trial, but is impaired, so they can't flex all the way, at least compared to the neurologically intact participant. So that's just for the thumb. If we look at some other joints, we can see some other patterns. So for example, for the elbow extension, so when they're doing this task, they extend their elbow about 40 degrees. All of these stroke participants have impaired elbow extension, so they're all below that baseline. If we look then at the shoulder, we can see kind of another pattern emerge, which is that this participant actually has overcompensation in the shoulder add and abduction. This participant really doesn't have much shoulder add and abduction at all, and this one is actually looking normal. So this is a lot of data, and these are only 3 of the 20 or so joints that you could monitor from the hand and the upper arm. But the point here is that if we care a lot about trying to understand how to give therapeutics and understand which pathways are damaged, we need to understand at least this level of detail to be able to say, okay, maybe this participant, yeah, they're a 38 and they have a subcortical stroke, but their damage is really in this pathway. This participant, their damage might be in this pathway, and this participant, their damage might be there. And I just want to highlight that this is not just a neuroimaging problem. It's not as though we just need better scanners that can dial in exactly what pathways are damaged. We also, after brain injury, we know that there's compensation by other networks. And so it's possible that even though there are pathways that are still present and still active, those pathways aren't strengthening enough to take over control from some of the damaged networks. And so it's both a matter of imaging as well as physiology and understanding what neural patterns and what types of brain activity can actually be generated. So towards this problem, our lab is starting a couple of projects to really get at this. So we really want to understand what are the movement impairments that we see in the population and how do they compare to the non-impaired population. So we have a couple of approaches that we're developing. One is a computer vision-based approach, so taking smartphone videos of people doing a very standardized task and using pose extraction methods to be able to infer joint kinematics. So this is a method that's actually from EECS here, from Jitendra Malik's lab, the hammer pose extraction algorithm. And that's something that we're hoping will allow us to very quickly build a large data set of intact participant joint kinematics. So we can start to see when we get an impaired person, are they outside distribution for certain types of joint movements, or are they within the normal variation we would expect? We're also developing a type of flexible, wearable, multimodal electronic skins that allow us to do long-term monitoring from not many patients, but some fewer patients, to really be able to see how are people using their hands and their movements in everyday life, how does it differ from their affected hand to their unaffected hand. And I'll actually be talking a little bit more about this in the future and how we can actually leverage this platform to even do interventional therapeutics as well. So this is one of our first kind of attempts of the lab at trying to understand successful behavior. So what does actually successful behavior look like, and what are the impairments we see, and can we develop a language that allows us to really understand where the impairments are in people with movement impairments? So I'm now going to transition to talking about some work in how the brain controls movement successfully. So what is a model of excellent movement control, and how does that break down in disease and movement disorders? So before I jump in, I'm just going to define a couple of terms. So I'm largely going to be talking about neural signals that are recorded invasively through local field potentials and single unit action potentials. So local field potential recordings are these low-pass filtered versions of voltage signals that you can record inside the brain. And generally, they're thought to reflect these synchronized firing of populations of neurons within a scale of about 1 millimeter. Single unit action potentials are when you've identified a unit, putatively a neuron, and it gives you a time series of zeros and ones, where ones indicate when the neuron is actually spiking and transferring information, zeros when that neuron is quiet. And these reflect very small spatial scales. So when I started my PhD, I was really interested in thinking about how the brain does control movement. And I was looking for a problem to work on, and I became really interested in beta band oscillations. So this is a signature that you can see by eye in the local field potential. I've highlighted it here in red. And it's this oscillatory burst that it happens with 3 to 4 cycles, typically lasts about 100 to 200 milliseconds, and it occurs in the LFP. And I became interested in it because it's known that it's exacerbated in Parkinsonian patients. And so I was really interested in how it might be contributing or blocking execution of good movement. So what was known at the time was that beta band power falls during movement and tends to be high before and after movement. But all of this work was fairly correlational. And so what I was interested in is, can I test the relationship between beta and movement onset in a more causal way? So I developed a task that was performed in non-human primates where they basically had to control in real time the amplitude of their beta oscillations. So I'll show you a video in a second, but they'll see on a screen a cursor that indicates the level of their beta amplitude. And when it's really high, they'll see the cursor go up. When it's low, the cursor will come down. And their goal was to actually regulate their own beta oscillations and then immediately afterwards perform a movement. So this is what the task looks like here. So here they, put their arm in the middle. Now, the cursor that you see moving is their beta power in real time, and their goal on this trial is to move it to a low state, and then as they get it into the low state, they execute a movement immediately afterwards. And so what this allows us to ask is, how does beta state affect movement initiation? And so what we see here is if we align to the time that the animals are able to move, we see that when trials are at that high beta state, movement initiation is a lot slower than when trials are at this low beta state.
This is an animal. How do I tell the animal that it should control move my curve into a certain point?
Yeah, like how do you train them to do this?
Yes.
Is that what you're asking?
You can tell a person, try to reduce your heart rate and I give you feedback, but I've never seen that done in cats. So how do you do that in animals?
Yeah, these sorts of biofeedback experiments are done all the time, but you have rewards that you give them for intermediate goals, like getting their, they can track cursors on screens, they know where target locations are, and then you give them juiced or food rewards for succeeding, and you can make the task, one of the things I didn't show is you can make the task easier or harder depending on how much they have to move that to get it into the target. So you start with an easier version, and then by accident they end up in... Sorry? These are non-human primates, so rhesus macaques, yeah. So what we were able to find is that high beta states delay movement onset, and we did find that we became really interested in, can this actually translate to Parkinsonian patients? So this is a patient population where there's too much beta. Can we use a neurofeedback system to see if they can actually improve their movement onset by doing this sort of neurofeedback game? And so we were able to build a system in collaboration with Medtronic and the Activa PC+S deep brain stimulator system. And we were able to show, yes, indeed, in these Parkinsonian patients, when their beta power decreased, they had faster movement onset times. And so now thinking about what does that imply for our models of excellent control of movement is that we know now that we kind of have to reduce this high beta state in order to execute movement well. So in my postdoc, I then became really interested in trying to better understand how neural signals are impaired in movement, when there is impaired movement, and how they naturally recover in the brain as movement gets better and better. That actually gives us a really nice system where we're looking at the same animal, often the same neural recordings, and we see impaired movement at the beginning, and the brain's natural recovery system allows us to track what the brain itself is doing to repair its own movement. And so in this system, we were looking at reach to grasp behaviors. We had a model of stroke that allowed us to give us impaired movement. So we created a small lesion in motor cortex that impaired the hand and upper arm function, and we recorded signals in adjacent motor cortex that we knew were important for reconstituting movement. So this area right next to motor cortex, premotor cortex, has been shown to be really important for recovery for movement after stroke. So just like that video that I showed you from the UCSF data, early on, movement was impaired, as you see here. There was a lot of variability from trial to trial. Later on, movement is recovered and is basically indistinguishable from the pre-stroke. So then we were able to ask about the same signal, which are beta dynamics during the recovery of these reach to grasp movements. So this is a trial, a behavioral trial from an impaired animal. What I'm showing you here in the black trace is the animal's movement speed. So you can see that they have a reaching event, a grasping event, and then multiple grasping attempts, but because they're impaired, they actually aren't able to grasp successfully. And what we can do is we can look at the neural signals in cortex and actually also in subcortex in these animals during this behavior. So what I've done to these local field potential signals is I've filtered them within the beta band, and I've actually highlighted in red where these beta bursts tend to occur. So all of these red periods here are synchronous beta bursts across multiple channels of recording in cortex. And so what I hope you can appreciate is that these bursts are very synchronous, both across cortex and subcortex. And when I show you the recovered state, you're going to see a very different picture. which is that these beta bands oscillations are actually now a lot less synchronous across cortex and subcortex. And so what we were able to find, to our knowledge for the first time, is that there are these two separable types of these beta events. So the first is these global bursts that involve a lot of channels, including cortex and subcortex, all oscillating together. And we find these in the impaired animals, especially during movement, which is very unusual. Again, remember our previous study was that these events rarely happen during movement and actually impair movement onset. And then in the recovered state, we actually see a lot of these much more localized beta events occurring in spatially clustered segments, so spatially clustered channels next to each other. And they do also happen during movement, but they seem to have really different dynamics with recovery. So we find actually that these global events go down as animals recover. So the x-axis here is a recovery index, and these global events go down. And as the animals recover, we actually see more of these local events occurring. And so we can refine our statement slightly to say not only are we overcoming high beta states, but we're specifically need to suppress these global beta states, but allow these local beta states to execute excellent movement. So the last kind of model building that I'm going to talk to you about today is based on spiking activity.
So you before mentioned that there was a question of causation, there is correlation. How do you know this is not just correlation?
Yeah, so in the second study, we definitely know this is correlation. In the first neurofeedback study, our argument was that since the animals are directly controlling their beta power, and we force them to kind of increase beta power and decrease beta power and then execute a movement, that's like an internal perturbation. And so I didn't show this, but actually if you look at when they increase beta power and they execute a movement afterwards, you see that the beta power trajectory has to come down for them to actually allow for that movement to be executed. And so it's very unlikely, in our view, since it's all within a same task, that beta power is indicating something else like attention or visual processing or all these other external variables because they're directly controlling that variable. So yeah, the final part of this model building will be looking at the spiking dynamics. So beta oscillations are a really nice signal that it's great that you can detect them actually non-invasively, but one of the things you don't know is exactly what are they saying about the spiking activity, which is the thing that's actually driving movement. And so we wanted to zoom into the very fine spatial scale and look at population neural activity recordings. And so this is an example of a number of the neurons that we were able to record. This is an example of the activity of one neuron. On the x-axis is time aligned to movement onset, and each row is the activity of a neuron, that same neuron during a trial. If we average across trials, we can represent this trial average trace of this neuron. And if we zoom out even further, we can represent spatiotemporal activity using a template like this, where the x-axis is time, each row is a different unit, and the color represents the activity trace that you're seeing here. And so with these trial average templates of spiking activity, we can ask a few different questions. So the first thing that we asked was about the consistency of these patterns. So how consistent are these trial average templates during early recovery and late recovery when we compare them to individual trials? So what we see here is if we look at a single trial, it's actually quite different than the trial average template. But if we look in the recovered state here, the single trials actually have a lot of the same signatures that you see in the trial average template, specifically this kind of sequential structure of spiking activations. And so we can quantify that using a correlation coefficient metric. And the main thing that we find is as behavior gets better, so as the animals are getting faster and more consistent, their neural activity is also getting faster and more consistent. And so I don't think this is necessarily surprising, but it is a hallmark of excellent control, is being able to reliably evoke the same neural activity pattern on trial-to-trial basis. And so we can add to this and we can say we have consistent spatiotemporal spiking. And then finally, we can actually look at the details of the spatiotemporal activity pattern. So not just the consistency of it, but the properties of it. And so one thing that you can appreciate is if you look at these different patterns, there's actually different types of bursting that you see. So there's really just one big burst in the impaired state, and then multiple bursts that you see in the recovered state. And so we can quantify a higher dimensionality with recovery. And then finally, we can also quantify this temporal predictability metric as well. And so the idea here is that although we know that individual trials are consistent, we also want to know whether at a given time point you can make a prediction about the next activity pattern. So the notion of a stable recovered network is that if you have one activity pattern, it will reliably transition to a predictable next activity pattern on a time point to time point basis. And so what we find is that indeed in the recovered animals, we do see very consistent temporal dynamics as well. That was a lot about what sort of structures we see in both the field potential and spiking activity. And so I'm just going to take a second to point out that all of this has been within motor cortex. And I really motivated this talk by saying that stroke is a very complex problem and is not just related to one particular area. And so I think of our work in motor cortex as really a focus on the cortical spinal tract. So motor cortex is a center that sends commands down the cortical spinal tract to animate musculature. And so if we understand the dynamics in motor cortex, we can understand what is being sent to the output. But we know that motor cortex is not an isolated entity on its own, and of course receives input from other cortical areas and subcortical areas as well. And so the future work in our group is really trying to dive into cortico-cortico communication, specifically between sensory and motor areas, as well as subcortico-cortico communication. And so I'll explain a little bit more what we're going after here. So we know that motor cortex has this privileged control, particularly of dexterous hand function, but we know that there are these other inputs that are also critical for control of hand function. And so this is a super simple video, but I think it illustrates the point really clearly, which is that a dexterous task, for example, picking up a match and striking it, is something that we can do super easily. So I believe the amount of time it took that participant was about 7 seconds to do that task. And now if we numb the digits, the cutaneous parts of the digits, we can see that this participant is going to have a little bit more difficulty. So note that they can still see the object. They can still make the same plan. It's not as they're confused about the objective of the task, but they can barely pick it up. They somehow manage to pick it up, and now they have this really complex task of striking it with exactly the right amount of pressure on the sandpaper. And you can see that they managed to do it, but it takes approximately 5 times as long. And so I don't think anyone is confused or has doubts about the importance of somatosensation, but I think this illustrates it really clearly that it's really critical to get inputs into motor cortex from these other areas in order to generate control. There's also literature describing how recovery is worse when there's damage to somatosensory cortex, so recovery from a stroke, and there's an inability to leverage intact cortical spinal tract pathways if somatosensory cortex is damaged. So it almost prevents the animal from taking advantage of some of the pathways that are there and do exist. We're also really interested in thalamic input into motor cortex. There's been recent literature illustrating how thalamus can shape the dynamics within motor cortex. And thalamus actually conveys a lot of information from both the basal ganglia and the cerebellum through a very narrow reduction in number of neurons and expansion into motor cortex as well. And so those are some of the tasks that are some of the brain areas we're going after. And the way that we're going after this is we're developing new dexterous tasks to study in the lab. And so just one example is this object manipulation task that we're having people right now doing, and eventually we'll be having animals doing as well, where the animals are pinching onto an object, manipulating it through a maze, and using the somatosensory feedback of the object contact with its environment to learn and refine their movements. And so we're really zooming into individual contact events to study somatosensory motor inputs. So that's what we're doing in the future. And now I've told you a lot about systems neuroscience, and I'm going to switch gears to talking more about engineering therapeutics. So we're going to take advantage of some of what we've learned about excellent control in motor cortex and try and design therapies based on that. So I told you a lot about high-dimensional, temporally predictable spiking, and so we can actually build a model that recapitulates many of these features. So this is a super simple model. This is a feed-forward SynFire chain, and I'm going to just show you what the activity of this network looks like here. So the x-axis, again, is time. Each row here is a neuron within this really simple feed-forward model. And the main idea here is that there is an input packet of neurons, and that propagates throughout the network to create this pattern. And so this is not a network that was trained by any means on the neural data or is designed to recapitulate features of the neural data, except that it has a very specific pattern. We know what that pattern is, and its goal is to run to completion. So very simplification of the system. And so we were actually able to create an impaired system and a recovered system. And we apply the same data analysis metrics that we used in the real data on the model, and we can find that indeed the recovered system shows an increase in template match, an increase in dimensionality, and an increase in predictability. So this is, again, a super simplification of the model, but we do have a system now where we can start to do experiments where we say, okay, we know what the recovered and intact system looks like. We know what the impaired system looks like. Are there ways of designing neurostimulation to make the impaired system look more like the recovered system? And so in my postdoc lab, there was a type of electrical stimulation that was being developed while I was there, which was called epidural direct current stimulation. So this is where electrodes were placed on the surface of the brain and small amounts of currents were passed between these electrodes to create these electric fields. And so the electric fields had the effect of slightly biasing the timing of spiking without directly activating neurons. So this is an example of neural data recorded when there's no stimulation. So x-axis is time, each row is a unit. You can see there's not very much structure in this neural data here, but as soon as stimulation was turned on, you start to see more coherent bursts of spiking occurring. And so this was really attractive to us for a couple of reasons. And one of them is a detail about the model that I haven't really explained. So in the model, this packet of inputs here, the statistics of how tightly coordinated those inputs are have a lot of determination on how far this neural activity will propagate throughout the network. So if these inputs here are quite spread out, that packet kind of dies out over time because each neuron in the subsequent layer receives a more spatially dispersed set of inputs. And so it doesn't always necessarily reach firing threshold. Whereas if this tight packet of inputs is really tight and happens all at the same time, then at each subsequent layer, neurons are receiving sets of inputs that are really tightly coordinated in time, and so it more reliably drives inputs, and you get further propagation throughout the network. And so we care about this because it's possible then that we can use this type of stimulation that helps increase coherence of neural activity to actually improve the inputs. And so the idea here is that if we can design a type of stimulation where it improves coherence of inputs, we can actually improve propagation throughout the network. So this is all nice in theory, but does this actually work in practice? So we did the experiment where we created a lesion, a small lesion in our animals. We implanted these epidural stimulation electrodes, and we delivered alternating current stimulation at 3 Hertz. And I'm happy to talk more about why we chose that waveform, but for now, it was our way of creating these brief periods of excitability. This created these electric fields within the brain, and we were able to see that indeed, if we look at single neurons, they tended to have preferred firing at specific phases of the stimulation. But the main thing we were interested in is could this actually enhance performance, potentially through improving propagation throughout the network? And so I'm going to show you just a video of the animal's behavior without and with stimulation. So just to orient you, there's a pellet here that the animals are trying to pull out of a well. So this is a dexterous task. So this is an animal with an impairment. So you're going to see that it takes them a little while to pull that pellet out of the well, but they do manage to do so. And then on the same day, with the same session, and the same animal and the same task, we now applied stimulation. And so the video on top is exactly the same as the last slide, so you can focus on the one on the bottom, but you can see kind of the difference here. And so you see much faster performance, much more feed forward, much more smooth execution of that movement. And so this does work. I've only showed you a couple of videos, but we tested this across multiple animals, multiple sessions, 2 different types of object manipulation and dexterous movement tasks, and we find that this type of stimulation was very successful. And so this was really exciting for us. This is a fairly minimally invasive way of delivering stimulation to the brain, and it was able to create these pockets of excitability that allowed the motor system to gain functionality that it didn't have before. And so some of the details of the stimulation I think are worth mentioning and are also directions of our work in the future. So when there's no stimulation, you had an activity pattern that only propagated partially. When you applied the stimulation, if the stimulation phase did not quite align with the movement, the beginning of the activity pattern, you actually did not see an improvement in the propagation here. However, if you did get a nice alignment between the stimulation phase and the input, you got excellent propagation throughout the network. And so you can imagine that this sort of phase dependence is not very desirable for a neural interface. You don't want to be walking around with your neural interface technology and only have it work half the time depending on when you happen to start your movement. And so there are a couple of approaches that we're working on to improve this. So one is we can actually do waveform optimization. So this is a very weak electric field, and the effect of the electric field is actually having these very slow depolarizing waves. So the 3 Hertz and low frequencies in general are really beneficial. high frequencies happen so fast that the neurons don't actually have enough time to integrate input. And so one thing that we tried doing is coupling the beneficial low frequency depolarizing phase with the high frequency phase that would just allow the waveform to stay charge balanced. And we actually see that we can reduce the phase dependency of the stimulation based on this. So this is one of those stimulation phases that are not particularly good for execution. And then we replace that with this new optimized waveform, and we actually see much better execution. And so this is kind of one trick that we've thought of for improving performance of the stimulation. A second is that the stimulation is actually being applied kind of right now, regardless of any understanding of the underlying neural dynamics. And so we have no idea what the underlying brain state is doing. We're just kind of overriding it with stimulation. And so of course, a lot of us are engineers, and so we say, okay, let's put a sensor in there and try and detect the underlying brain state and try and actually deliver stimulation that's optimized to help the brain at that particular point in time. And so right now we're working on systems to be able to do this type of detection while we stimulate at not just 3 hertz, but maybe at a randomized frequency that's matched best to the waveforms that we're detecting. One specific goal that we have is these localized beta bursts that I mentioned before. We actually hypothesize that these are communication bursts between cortico-cortico sites. And so one possibility is that if we detect these bursts in, say, a prefrontal cortex or somatosensory cortex, we might be able to then help write that burst into a motor cortex in a set of localized channels here to enhance cortico-cortico communication underlying dexterity. So that's one particular application that we're thinking about. So future work will then be trying to optimize these waveforms and designing closed-loop systems to make this type of stimulation more responsive. So with the little time I have, I'm just going to talk about one last project. And so one of the things that we've made a number of assumptions about the patient condition when we try and develop this type of stimulation. So one assumption is that the neural network is movement capable, meaning that there's actually a really good output from cortex to the spinal cord, and we just have to fix activity up in cortex, and it'll magically get to the spinal cord into the musculature. The second is that we've assumed that the right neural patterns for a given movement have already been formed, so that the participant actually already knows how to execute the right neural patterns for a reach to grasp task. We just kind of have to help the brain do them. And then the final is that full propagation of these patterns will actually yield fast and smooth movements. And so I would argue that I think this is true for a subset of patients, these minor to moderate stroke patients, and that's about 70% of the patient population. And there's actually work ongoing in Rich Ivory's lab and the psych department on trying to use non-invasive stimulation very much modeled after what we have right now. And they've been able to show improvement in performance in stroke patients with this 3 hertz stimulation. And so we have good reason to believe that this will actually work in people that can move and don't have super large deficits. However, what about the severely impaired stroke patients? So there are a subset of patients that are closer to paralyzed. They really can't move at all. They're kind of stuck in, you may have seen them in a very typical stroke posture state, so flexed elbow, flexed wrist, flexed fingers, and then very difficult to make much movement after that. So is there, what can we develop for that type of participant? And so it's very likely that the neural network there is not movement capable. We have no idea if the right neural patterns have actually been formed for the movement, and we don't know if full propagation of patterns will yield fast and successful movements. It seems quite unlikely. So one thing that many of these participants have, though, is they still do have cortical-spinal tract connectivity to the output. So this is an example participant that I've been able to work with that has a very large lesion and quite a bit of, like a very low ARAT score. She's close to a 0. But she still has these seeds here that you see in red and green, which are connectivity from the spinal cord up to cortex. And so we know that there's still pathways of output. And we know that this participant can actually create muscle activity. So it's not suprathreshold, it's not enough muscle activity to be able to move the arm, but it's subthreshold, so it's something that we can start to give biofeedback on. And so without getting too much into the details, myself and many other collaborators, part of a large team here, have developed a type of invasive neural interface therapy, which tries to leverage the fact that this type of participant actually has connectivity to the output. So we implanted an array in parts of the brain that still had cortical spinal tract connectivity, and we built a brain-machine interface decoder that then moved an exoskeleton of this participant's arm. And so the goal here was to really give feedback about the neural patterns in premotor cortex that still have intact descending tract connectivity, and to try and reinforce trial-to-trial consistency like we found in our previous study as an important feature in these neural patterns. once the participant was really good at controlling the exoskeleton with their brain, we then started to add muscle signals into the decoder. So the idea here is that we actually know quite a bit about what muscle signals are important for, or what features of muscle signals need to exist in order to make the arm move. For example, if you have a hand that's opening, you can't activate your extensors and flexors to the same magnitude, otherwise you'll never be able to open the hand. You need to have an imbalance between them to actually create motion. And so we were able to build what I'll call a biomimetic decoder, where we take these muscle signals and map them into movement of the exoskeleton. And so the goal here is that, although it's a complex system, we are trying to reinforce co-activation of these premotor cortical patterns and biomimetic muscle patterns to drive rehabilitation. So one participant has gone through this study. I'm going to skip kind of to the main point just for time, which is that we've been doing this trial for up to 4, now almost 5 years, and we have seen unprecedented recovery for this level of participant, so someone who is this impaired. So this is another clinical scale I haven't talked about before, but another one of these 0 to 66, and you get one number. But this is a clinical scale that specifically measures impairment. And so our participants started here, and throughout the 4 to 5 years, they've steadily grown and shown improvements in impairment. And actually, if we compare that to other studies that are kind of state-of-the-art for this type of patient population that are super impaired, we see that we've been able to get improvements in Fugl-Myers that almost is double what some of these other studies are. And the main thing that I think is really important is that these studies tend to take months, maybe even just weeks, but we've been working with this participant for years and years and years, and this study, as well as others, have kind of combined to report that therapy dosage is really important. Being able to rehab, being able to continue to work all the time is something that's really important for being able to impart improvements. And so with that, this is an ongoing trial. Happy to answer any questions about the details of that. But the main thing that I think for us as a main takeaway is, are there ways that we can actually give participants, not necessarily brain implants, but ways to continue to do therapy at home in their own time, low cost, so that they can continue to match these therapy intensities. And so there have been some studies that have shown that just muscle biofeedback can be really effective over time. So these are examples of muscle signals from impaired patients right before a type of muscle intervention and then after a muscle feedback intervention. So you can see that they've gained quite a bit of volitional control. And they're actually able to increase their kind of extensor ratio, which is extensors over flexors. The main point being that you can reinforce specific types of muscle activation. But there are a lot of pain points to these types of studies. There's a lot of equipment. The electrodes to place on the arm are super bulky. The wires are bulky. The placement from day to day is something that's really variable, and those are important things to try and optimize and keep very consistent if you're especially trying to teach someone over years and years about their muscle signals. And so kind of looping back to one of the things I was talking about before, some of our technologies for developing these thin, flexible, wearable skins is the hope that we can actually create these stable interfaces for doing this sort of muscle biofeedback therapy at home. And then kind of create games. This was a game from an example paper where, their extensor ratio moved a skee ball and then they got points based on how much they moved it. So making these games really engaging as well. And so this is something that I'm really excited about to start launching. There's amazing groups here in physical electronics that work on thin, flexible sensors. And so we've been partnering with them to try and develop applications for their technologies. And so with that, these are some of the work that we've done in advancing models and engineering therapies. And if we go back to this technology explosion, my hope is really that with time, we're actually able to leverage this, both the sensing technologies as well as the neural interface technologies, to really be able to meaningfully make suggestions for patients depending on their specific impairments. And so for time, I'll stop there and thank both my postdoc lab, my PhD lab, the ISMOR trial, which was, as you can imagine, probably a huge group of clinicians, engineers, neurologists, scientists, physical therapists as well. And then finally, my new lab here at Berkeley, who's helped support an enormous amount of this work. So thank you very much, and be happy to answer any questions.
Okay, questions?
Hi. So you mentioned that kind of part of the reason you're developing these systems neuroscience models is to be able to fill in normal models of movement for people that are impaired.
Yeah.
How do you handle patient-to-patient variability in those kinds of models, or like how do you trade off generalizability versus like making your model specific to the patient?
Yeah, right, because we started with the notion that this is just a really complex syndrome. And so my thought is that within the preclinical models, we have actually a lot of control. And so, for example, the lesions that we did in these animals were all very consistent. And so they gave us a sense of what happens when we lesion motor cortex or when we lesion the cortical spinal tract, what sorts of signals are missing there. And so my thought is that we can start to piece together kind of from these preclinical models notions of how individualized pathways contribute to lacks of certain neural signals or excess of certain neural signals. And then when we go to the patient, we can kind of reverse map that and say what combinations of impairments might they have, or what combinations of neural signals might they have. Because the reality is, it's also very difficult to get invasive neural recordings from patients. So a lot of this work is going to be grounded in the preclinical model. Yeah.
Could you go back?
Can you do that?
Oh, sure. Could you go back to the graph of that showed someone's
Oh, sure.
their improvement over like multiple years?
Yeah, this?
Yeah, I was curious. So on the left, like that big first
Yeah.
that big first jump. Like, do you have any explanation for why you saw so much improvement?
Yeah, so this is a participant that had their stroke like 10 years prior to this intervention and had tried rehab at the time of recovery, but there was a big gap between any sort of interventions and what we started. And so, yeah, I think a lot of this is just the intensity, like especially this early phase. We probably were rehabbing about 4 to 5 hours, 5 days a week. Here we started to taper off into more like 3 days a week. But I think one other thing to point out is the difference between these different scales. So this is an impairment measure. So what that means is like the Fugl-Meyer, they actually ask you to do things like put your arm behind your head. It's not really a functional task, but it says, how can you actually do this? Can you extend your arm and do this range of motion? And so what we found is that as soon as this impairment increased, it then took the participant some time to start to integrate that improvement in impairment into actual functional tasks. So now that I can move my arm more, what can I actually do with my arm that's functional? Oh, great, I can turn on the faucet. I can use it to support vegetables while I'm cutting with my other hand. And so I think that's, I know that's not your question, but the difference between these plots really is about, the uncovering of impairment and then the integration of it into everyday activities.
That's really helpful. Thank you.
Is the noise in that left-hand plot noise, or are those actual real changes?
I think they're noise. This is towards your question. Like both of these are clinician viewed scales, and there's also, in this patient in particular, and I think this happens with a lot of patients, a lot of variability in their condition from day to day based on things we have no idea about: mood, sleep, general motivation. And so, unfortunately, we actually took clinical scales like a number of days in a row. And it was also very tiring. So kind of the ordering of the clinical scales also adds some noise. I think it's actually really nice to be able to see what that noise level is. A lot of trials don't have this many measurements. So, yeah.
That was a really great talk, Priya. I have a science question and an engineering question. My science question is, do we know, because I certainly don't, why this beta band activity is so important for impairing or facilitating motor movements? Like, do we know what its role is in the neural circuit? And then my engineering question is, the intervention you have now goes below the skull. Could you actually use over the skull TDCS to actually help improve motor movements?
Yep. So the first question is about beta band oscillations. So I didn't show this here, but in some of our data, we were able to record single unit activity at the same time as beta. And one thing that we see is during global beta, if we look in motor cortex, we see neurons that are super entrained to that global beta. And if you look at the firing rates of the neurons and the firing rate variation, it almost clamps it. So the firing rate stays about the same, but if you look at the amount of variation that's possible with that neuron within a beta oscillation, it's just much lower standard deviation than what you would expect outside of a beta band activity. If you look at the local beta band, those are much smaller impact events, and so you don't really see that effect. And so what I think is that when, particularly in impaired states, I actually have a plot. I didn't put it in the slide. But in impaired states, we have neural activity patterns very similar to the ones I showed you, the spatiotemporal patterns where you have kind of one burst, then the next, then the next. And so when there's impairment, especially in grasping or hand function, you get a drop off in that excitation or that modulation. And so what I think happens is those global beta bursts kind of fill in that spot when cortex is not very excited. And so I think it's a default state that kind of is helping hold the current firing rates as they are. And when there's not enough driving movement or not enough very strong modulation that you tend to see with reaching, grasping in normal, healthy states, you get kind of just like the filling in of that void with beta. So I think it's a marker. I would never say it's like a causal driver of the impairment, but I think it reflects that kind of non-modulated state. And then your second question was about, can you use non-invasive methods to? Yeah, so one thing that we found is, in our experiments, the electric field that was required to actually bias the neurons was close to about 1 volt per millivolt per millimeter. And so we were able to get that with the electrodes on the surface. TDCS, as well as like TMS, is also another way you can get very high electric fields, but it's very spatially local. So one thing that we think is important is the kind of spatial distribution of that field. And so right now, I don't think you can get that with TDCS. You tend to get like 0.2 millivolts per millimeter because a lot of that current is shunted by the skin and the muscles and the skull. But yeah, if you're familiar with Rich Ivory's KTMP type of stimulation, that's actually something that is like TMS, but has the ability to create this modulation of like 3 hertz or 10 hertz or 20 hertz on top of it. Typical TMS just delivers single pulses. And so this is a new technology that we're really excited about that might be able to impose this sort of kind of slow moving bursts of excitability. The only thing I would be a bit skeptical about is it still is very spatially focal, this KTMP. And so I think we don't quite understand exactly how big of a volume you really need to be stimulating to get this effect.
Is the KTMP something that people are going to be able to use like all the long term?
Yeah, right now it's very much like come to the clinic, get this
'Cause
intervention.
TMS is usually a focal thing.
Yeah, there's maybe you're familiar with this, but there's this one company that this like Motif neuroscience company. So this is a device that can be implanted just in the skull. It's a passive device, and then it's powered and has electronic transmission from like a wearable hat. And so this is also kind of the same style of like minimally invasive, but it's supposed to be TMS-like and something that people can kind of bring around with them every day. So I think technology will eventually get there if it proves to be successful.
Yeah, very cool talk.
Thanks,
I had
Jay.
a question about when you were doing like the simulation waveforms for the
Yeah.
monkeys to improve kind of the feed-forward path. Did you use like the same simulation patterns for each monkey? And if so, do you think you would see like a significant improvement if you tried to optimize it on like a per like patient basis?
Yeah, that's a great question. So we stuck with 3 hertz, so it was our favorite number. But the reason we chose it is actually because the animals' movements tended to be between 300 and 500 milliseconds long when they were successful. And so it was really matched to like the frequency of their movement kinematics. And in general, we know that movements tend to occur in this 1 to 3 hertz range if you look at sub-movements or the statistics of grasping, reaching, all of these sorts of movements, they're in this low frequency range. And so that's why we chose it, because we thought that we were going to be matching kind of movement onset as that was our best chance of matching movement onset as regularly as possible. I think in terms of optimization for individuals, certainly there are individuals who move faster and slower, and so I think there could be room for optimization. But I think the more important point is getting the alignment between that frequency and the movement onset right. I think that's a much more important factor, and that would require kind of sensing on the individual timescale. Yeah.
Gotcha, thanks.
Yeah.
Questions? I have one.
Oh.
Okay. So I also have a question on your 3 hertz sine wave.
Sure.
So when you went from this sine wave to sort of the more complicated form,
Yeah.
I have sort of 2 to 3 questions on that.
Okay.
A is, do we have any understanding what changes in the brain if you go from the sine wave to another one? Can we sort of see a little bit more causal what's the pathway, maybe even that pathway is to your hands, that it actually leads to better performance? And then the second is, again, similar. Can you adapt that form to the patient? Is it clear that all patients should have the same form? Or could I maybe use some machine learning algorithms that I learn on the patient with some, I don't know, reinforcement learning or something that, oh, for me, you should use this form, and for you, maybe the sign is just fine.
Yeah. Okay, so your first question was about what happens in the brain when you go from one sine wave to a second?
Yes.
Is that right? So like
From the sine wave to this nonsense.
to this. Yeah. So we're not sure, how like this works in theory, but we haven't tested this in the brain. The hypothesis is that you can kind of see it in the patterns here, which is that the sine wave creates these very smoothly varying kind of background increases in excitation or decreases in excitation. And you can kind of see that by these bands that become more darker when the sine wave is in its excitation state and lighter when it's in the more inhibitory state. And so what you get here is also what you can kind of see by eye, which is that the low frequency creates these darker bands that are very slow, and the high frequency creates these very narrow periods of inhibition. And in this model, the length of that period was short enough to allow this pattern to propagate over that white band without dying out or without losing any power. But that's something that I think we really don't know in the brain. We do know, for example, that neural activity when you're delivering, for example, step pulses, so you go from zero current to immediately turning on a current. We know that neurons really like that change in current and generally align to that. And then if you keep the step pulse on, you start to see kind of less synchronization of neural activity. And so it could be that, even our model is not accounting for the fact that even though these are very weak fields, this high frequency sine wave might actually be entraining neural activity more than the low frequency, just because it has kind of that steeper gradient of change in voltage. So that's something that's not modeled in here.
If that's true, maybe you would just do square waves.
Yeah, that could be possible. Yeah, and that's if you wanted to like really align neural activity very specifically. Generally, the experiments that I was referring to tend to be at much higher voltages, and so you're actually trying to directly excite axons or drive neurons. Here, our idea was very much that we wanted to bias neural activity enough so that the underlying patterns could unfold, and if the underlying patterns were very strong, we wouldn't really interfere with them, but if we could help boost them along, then our stimulation would be able to do that. So the approach has been to try to stay away from directly activating neural activity, because I would say we just don't know enough about what exactly the right patterns are. Could you remind me of your second question, please?
Whether there might be a way to inform to the patient.
Yeah. Yeah. This is very much based on a simplified model that the initial activation of the neural pattern is very important for propagation of the pattern through the network. But you could imagine patients, for example, that have excellent activation of the patterns and propagation that would go very far, but maybe somewhere in the middle, they actually have some deficits. And if you could only bridge that middle section, then you would have better luck with behavior. So I think maybe not, right now I don't have a very clear idea of how you would optimize frequency, but I think you could very much optimize timing of the stimulation waveform and where that excitatory burst happens to the behavior that the people are doing. With respect to frequency, it could also be that, if you're trying to optimize a very fast movement versus a very slow movement, those could have different frequencies and timescales, but, and that's potentially something that you could learn through machine learning. Maybe what you're referring to, though, is if you're just able to kind of deliver a lot of different sine waves to the brain and then have some metric that tells you this one was really great, let's kind of converge on that, more of like a search algorithm and then optimization there. I think that's possible, but I think you have to have a very good understanding of your behavior and of what your neural activity should look like in order to do that optimization.
You say the problem is then that I actually don't quite have a good optimization function because they have these very crude measurements whether you improve or not.
Yeah, that's right. Yeah.
Could you go to the network layer time map where it shows impaired patient to like the normal? Yeah. I can see like just in the background, like I can see the actual bands being formed on the very right, whereas like the one on the very left, like I don't see any sort of like bands in the background. Like, is that also like similar to like the waveform, like the optimized waveform?
Yeah, exactly. So this is no stimulation, so there's no nothing that is driving that for increased background excitability. But yeah, this is exactly the background excitability that is the effect. Yeah, and so it could be possible, for example, to make these narrower, make them wider as you change frequency, or shift where that band happens with respect to the start of the pattern. Yeah. Yeah.
Maybe one last question.
So when you're stimulating at these very low frequencies inside the cortex, are you
Yeah.
not worried about like nerve death or like charge buildup in those like regions of the brain?
No, we definitely are. Yeah, so this is, you mean because the low frequencies,
Yeah.
you're basically, like if you zoom in, it's basically DC.
You're blocking, yeah. Like nerve, you're blocking conduction, right?
Yeah, okay, so I guess two things. So one is we're not really worried about nerve block because these are such weak fields. So I think if we were to hyper boost up the field strength, I would start to be worried about things like epilepsy or like seizures or any sort of thing where you have super synchronous activity that just takes over. But this is so weak that we're barely entraining individual neurons. We're not even directly firing neurons. So I don't worry about that. What I thought you were asking was actually about on the electrode surface itself, if you worry about, putatively this is charge balanced, but it's charge balanced over a time scale of 300 milliseconds, which is very different than what we think of charge balance with conventional stimulation, which is on the order of 1 millisecond. And so that's actually, yeah, we've done some post-mortem work to look at the screws themselves and look at the electrodes, and we don't really see any like rusting or buildup of oxidation, but yeah, I would say this is one difference between conventional electrode stimulation that exists and is FDA approved and is in people, and this type of stimulation is that we're not sure exactly how safe this is over long periods of time because of exactly that problem. Yeah.
Okay, well, thank you again for this super exciting talk.
Thank you.